Good morning, students. In last session, we discussed about the cell organelle, that is the capsule or slime layer. Today we have to discuss the next cell organelle, that is the cell wall. Prokaryotic cell almost always are bounded by a fairly rigid and chemical complex structure present lies just outside the plasma membrane or in between the membrane and capsule, that is the slime layer called the cell wall. Okay, means the outermost covering of the cell, that is the slime layer. That means the organism which bears the capsule, for that the outermost layer, that is the slime layer or capsule layer. But the organism who do not bears the slime layer or capsule, such an organism, the cell wall is the outside layer of the cell. But the cells who bears the capsule, for these organism, beneath the capsule there is a cell wall, and that is just outside the plasma membrane. Okay, that means in between plasma membrane and capsule or slime layer there is a one protective layer, that layer which is chemically complex in nature and that is present in the cell of the bacteria, that is called as a cell wall. Okay, it is one of the most important prokaryotic structure for several reasons. Means whatever the functions, they are carried out by the cell wall, and due to which there is a importance to this structure, and specifically it helps to determine the shape of the cell. Means whatever the rod or cocci are the cells, that means that shape that is given due to the cell wall of the bacteria. Okay, it helps to protect the cell from osmotic lysis. It can protect the cell from toxic substances, and in pathogens it can contribute to the pathogenicity. Means if these organisms comes in contact with the toxic material, and these toxic materials, they are penetrated outside to the inside of the cell through this cell wall, but that cell wall, that is not allowed to pass such a toxic materials from outside to inside of the bacteria. And in some cases, that means specifically in pathogenic organisms, whatever the pathogenicity means ability to cause the infection, that is dependent upon the cell wall content, and that's why that cell wall plays an important role. Now the importance of the cell wall is reflected in the fact that relatively few prokaryotes lack the cell wall, and the best example of the organism who lacks the cell wall, that is the mycoplasma. In mycoplasma there is a cell wall is absent. Okay, but in other organisms cell wall that is present, and it will functions as a protective layer. Those that do have other features that fulfill cell wall functions. Okay, the prokaryotic cell wall also is the site of action for several antibiotics. Therefore it is important to understand the structure. That means when these organisms, they comes in contact with the antibiotics, and here antibiotic is a substance which is secreted by some microorganism, and that is inhibitory or it will kills these organism by that antibiotics. Okay. In antibiotics, again there are two types of antibiotics. One is a short spectrum antibiotic and another is a broad spectrum antibiotic. Short spectrum means either it will kills gram positive bacteria or either it will kills gram negative bacteria. That is called as a short spectrum antibiotics. But there are some broad spectrum antibiotics which will kill both gram positive as well as gram negative organism, and they specifically act on the cell wall of that bacteria. Is it okay? Therefore, it is important to understand the structure of the cell wall. Okay. Now, the peptidoglycan is an main component of the cell wall and is responsible for the shape and strength of the cell. Whatever the chemical composition that is present in the cell wall, among that there is one of the most important substance, that is the peptidoglycan, is the one of the most important component of the cell wall. Okay. It is a disaccharide. That peptidoglycan is a disaccharide and contains two sugar derivatives. It contains two sugar derivatives, that is the N-acetyl glucosamine and N-acetyl muramic acid, joined together by short peptide chains. Means here is the conjugated protein that is involved in the synthesis of cell wall. See, this is the structure of the peptidoglycan or composition of peptidoglycan. There is a two derivatives of, that is the carbohydrate, and that carbohydrate is nothing but the disaccharide. Okay. And to this disaccharide there is a peptide chain that is attached, and they form this type of structure, that is the here N-acetyl muramic acid, that is the yellow color. This is the one derivative of sugar, N-acetyl muramic acid, and here N-acetyl glucosamine, that is the brown color. That means these are the two derivatives of the carbohydrate. Okay. And here there is a peptide chain, that is globus structure or that coca-like structure which is seen here. That structure, that is called as a peptide chain. Okay. And here in between these peptide chain there is a pentaglycine interbridge that is present, and that bridge that is responsible to hold these different chains together. See here, here is the one interbridge and here is the another interbridge. That means the two chains they are hold together. Now here is the another interbridge which will hold the third chain, and it here it will the another pentaglycine interbridge, it will holds the fourth chain. In this way this structure of peptidoglycan that is formed, and that is one of the most important chemical substance which is present in the cell wall. Okay. Here N-acetyl muramic acid carries a tetrapeptide side chain. Tetra means 4, peptide means there is a bond between the two amino acids. Okay. So tetrapeptide side chain consisting of D and L amino acid. That means the dextrorotatory amino acids and levorotatory amino acid. Okay, see that D-glutamic acid and L-alanine, these are the amino acids that are involved in the formation of that peptidoglycan structure with mesodiaminopimelic acid, that is the gram negative bacteria, L-lysine, that is in gram positive bacteria. Okay, that means that mesodiaminopimelic acid that is present in gram negative bacteria, while the L-lysine that is present in the gram positive bacteria. Now tetrapeptide side chains are interconnected by pentaglycine bridge. We seen that there is a pentaglycine bridge and which will hold the side chains or the chains of these N-acetyl-muramic acid and N-acetyl glucosamine. Means these structures, they are held together by this pentaglycine bridge. Okay, and most gram negative cell walls lack interpeptide bridge. In case of gram negative bacteria, such a interpeptide bridge, that is, it lacks the interpeptide bridge. Cell wall provides shape to the cell and protects bacteria from changes in osmotic pressure within the bacterial cell, and it will measure 5 to 20 atmosphere. Okay. Now this bacterial cell can be classified into gram positive or gram negative based on the structural differences between gram positive and gram negative cells. There is one scientist, that is the Christian Gram, who developed the technique that is the gram staining, and based upon gram staining, whole population of bacteria that is get classified into two groups, that is the gram positive and gram negative. Okay, gram positive bacteria, they are stained by violet color, that are stained by violet color, and gram negative bacteria, they are stained by pink color. Okay, and this coloring to the cell wall, that is different. That means one cell wall or gram positive bacteria cell wall that will take the violet color, and gram negative bacteria that take the pink color. It is due to the differences in the chemical composition of the cell wall of these bacteria, and based upon this gram staining technique, the whole population of bacteria that is get classified into two groups. Okay, the cell wall of the gram positive bacteria have simple chemical structure compared to gram negative bacteria. That means the structure of the gram positive bacteria, that is the simpler or simple as compared to gram negative bacteria.